what is happening? They are all in circulation. So like I said, airborne conidia is entered. Even the SPA, A and D will try to attract these conidia and try to capture it. So this is a survey being done by all the PRRs. Collectins. So these are, the slide is just telling you what are the names. The APC can produce a lot of PRRs. So just remember a few names, the TLRs, CLRs, soluble pentraxins, complement. These receptors are very important for holding the PAMPs, attracting the PAMPs. Again, this is again node-like receptors. These are a lot of them which are now trying to survey a fungal polysaccharide, or it is a bacterial, or it is a protein. This is the soluble, again, they are taken from a textbook where they are trying to say that MBLs released from alveolar macrophages or SPA released from alveolar macrophages helps in immediate opsonization. So they have seen these are documented in pneumocystis or aspergillus, even in cryptococcus, etc. So these are very brief in action, soluble PRR. Finally, when there is an interaction between the phagocyte and the dendritic cell, the phagocyte is waiting there. So suppose a phagocyte has a dectin-1 and a TLR2 from a APC, dendritic cell, they will combine together, the phagocyte gets activated and there is phagocytic killing. This picture is just telling you by whatever other receptors, whether it is CR1, mannose, CD14, TLR4, dectin-1 and 2, all of them will lead to phagocytosis opsonization. This is where innate immunity is very robust and acting to kill the pathogen. Now, these cells are in circulation wherever or near the mucosa, wherever is the site of infection. Now, the site of infection is supposed at a distance, and you need all the circulation, the cells which are in circulation, the monocyte, the progenitor cells, the monocytes, the macrophages, to reach the site of infection. So these are a few slides telling you how these cells reach from blood circulation to the site of infection. So these are by adhesion molecules. These can come as short notes. What are adhesion molecules? Like integrins, short note on a selectin. These are present on all leukocytes. So all the cells which are capturing these fungi, they have adhesion molecules also. These molecules will adhere to the vascular endothelial cells. The vascular endothelial cells are then lead to aggregation of leukocytes by integrins and by ICAM and VCAM. Platelet aggregation molecule, capture adhesion molecules, or the ICAM molecules. These have the endothelial inter and endothelial adhesion molecules, and these are also platelet adhesion molecules. You see the next picture here, where if they are just rolling in the blood circulation, then they will never get attracted to the site of infection. So these adhesion molecules allow these cells to get aggregated, and then there is a diapedesis. So integrins are one of the major adhesion molecules which you must remember. Of these are ICAM and PCAM. And this is a picture where it will tell you that leukocytes are just rolling. If they are just rolling here and there, they will never undergo a diapedesis and reach the site. So these PCAM and ICAM, these molecules will cause adherence and then diapedesis, and then they reach the site of infection. So this is happening for all pathogens, whether fungal, bacterial, or even parasitic infection. This is a picture where, if you are asked, what are the steps occurring in a neutrophilic killing? So reactive oxygen species, just look at these steps: activation of phagocyte, phagolysosome fusion, degranulation. The proteases will release the microbial peptide into smaller parts, exposure of these components, digestion, and finally it is released, and apoptosis occurs of the neutrophil. So it kills the fungi. And this is one of the slides where it is telling you that once it is recognized by PRR, so this is the second arm, invasion of the fungus, PRR, a cytokine, suppose it is interleukin 23. 23 is a signature molecule for TH17. So TH17 get activated, IL-17 will be released. They will lead to acute inflammation and recruitment of more neutrophils towards the site of infection. Now comes the complement. Complements are bioactive molecules. These are proteins, again in the circulation. This is simple. It is they will have to attach to a phagocyte or a antibody. So either it is a classical pathway or it is an alternative or lectin pathway. So whichever is the cascade, they will cause opsonization and osmotic lysis. So this is how it occurs. A classical will be an immunoglobulin which has been released by the B cell. They will attach to so the basic component is C3A, C3B. Remember your old story. Then C5A converts into C5A and B. Whether it is lectin or alternate pathway. Lectin pathway, you see the MBL. You remember the receptor I showed you will get attached to MSP. MASP is a mannose adhesion, I think, serum protein. So this is a mannose protein from the fungi. This attaches to MBL, and then the lectin pathway finally, it forms a complex, the MAC, the membrane attack complex, which will kill the fungi. So this is what it is saying. Either it's an alternate pathway, classical will have immunoglobulin, or a lectin pathway. It will have inflammation. The complex will finally be the membrane attack complex, which will cause the lysis of the fungi and osmotic killing and opsonization, etc. Now let us come towards the acquired immunity, the dendritic cell. Let's look at the dendritic cell, which is one of the major player in this role, phagocytic cells. So they link the innate and the acquired immunity. So the DCs, I'll use the short form, the dendritic cell, they ingest the yeast very easily, and then they can induce, whether they will take it to a CD4 T cell or a CD8 T cell, and accordingly will activate whether it's a Th1 or a Th2 response. Now, the origin of the dendritic cells, it can be from the blood circulation, they can be from the bone marrow, they can be from dermal dendritic cell, Langerhans dendritic cells. So, wherever is the site of infection, whether it is GIT, the respiratory tract, the genitourinary tract, the dendritic cells are already present there and they get activated, and then they attach to the fungal protein and will take it towards the effector mechanism, that is to the T cell. Either they ingest themselves, as I said, because of defensins and etc., or if they are incapable of doing that, they will release cytokines, and in that cytokine milieu, they will find out whether it is, I need to attach to a CD4 or a CD8 or a B cell. Why DC is important? This is now, how do you, DC is important because they have co-stimulatory molecules. This is very important. Do you want a DC to capture a self-antigen and present it to the T cell? No. Then it will create a havoc, cause autoimmune disease. Co-stimulatory molecules are a signal where it stops the T cell from identifying the self-antigen. So if a co-stimulatory molecule is absent, then fungal infection can be fulminant, and the patient usually has invasive infection. So co-stimulatory molecules are the very important secondary signals to allow T cells to mature and activate. What are these? This slide is just telling you, if you want to identify the dendritic cell. Suppose you are doing a project, you want to see in Cryptococcus or in Pneumocystis, what is the population of dendritic cell in a patient who is undergoing chemotherapy. So these are the various markers. The markers will identify whether it's macrophages, the Langerhans cells, etc. So this is for flow cytometer to identify these cells. So DCs are very important for capturing. They are the first levels where they will also express the toll-like receptors. They also result in co-stimulatory signals, and their locations are those sites which I told you. They're mostly in the mucosal areas, and they are a critical step in T cell priming. Now, macrophages is another important cell which is found in circulation. So usually they are derived from monocyte. The monocytes are small in number. Macrophages can be either circulatory or it can be residential macrophage. Resident means at the lymph nodes or the alveol. Suppose it's near the alveolus, alveolar macrophages. Any lymph nodes, they are resident macrophages. Macrophages have a longer life than neutrophils, and they stay longer and cause a more robust phagocytic activity. And they are also involved in a lot of granuloma formation, like in a chronic infection. And macrophage also have a different, if you want to identify whether there is a macrophage activity, so their cytokines, what they express is very different. They usually GM-CSF or TGF-beta, these are the ones which they produce. So all cytokines are not expressed by all cells. So when you go through the list, you will see, GM-CSF is maximally expressed by the macrophages. So they are the important defense again, Candida, Aspergillus, and Mucorales, and they are evident by causing severe granulomatous reaction. So resident macrophages can kill the pathogens, and more monocytes can get recruited and form macrophages, resident macrophages. They release chemokines and they can attract more monocytes to the sites. And this is what I was saying, interleukin 1, 6, TNF, they live longer, and this enhances antifungal activity. The IFN, GM-CSF, colony stimulating factor, and the granulocyte colony stimulating factor. In fact, this is given as a therapy in case of bone transplant patients to enhance the macrophage activity. So this is how from bone marrow release into circulation activated macrophages, and they also go into the resident macrophages. So this cycle, this is a very beautiful slide where you can see that these monocytes have two edged work. One is inflammatory. So the immediate reaction that I have encountered an antigen, my reaction is inflammatory. I will produce a Th1 or a Th17 and classically cause a inflammation and reduce the infection. Whereas the immunomodulatory dendritic cell, these have a different kind of an activity. They are the ones which are bringing down the inflammation. So their role is to bring a balance. If immunomodulatory dendritic cell becomes haywire, suppose they become excessive in nature, then the fungi gets a chance to persist. So you see there is a very thin line of difference. An inflammatory dendritic cell will release inflammatory cytokines to bring down the infection. Anti-inflammatory immunomodulatory dendritic cell is trying to curb down an inflammation. Why? So that it does not disturb the tissues. The tissue damage does not occur. However, if suppose by chance the activation is so much that the immunomodulatory dendritic cells are excessively produced, then it is like an anti-inflammatory response. The fungi which have been left loose, they have not been attacked, they find an area and an opportunity to remain in the circulation or at the site of infection and try to continue multiplying. So these pathways promote the development of, actually immunomodulatory cells promote the development of T regulatory cells, which are you can see the T regulatory because of IL-10 or a Th2 response. So if they are in a balanced number, then Th1, the immune response will be curbed and infection will settle. But if the balance of this immunoregulatory pathway will determine whether a persistent infection results in a chronic immune-mediated tissue destruction or a protective tolerance. So this is very important. Either it causes a tolerance, so the fungi remains, or it will cause a immune-mediated tissue destruction. So immunomodulatory, remember there are two ways. One is CTL4 and one is PD1. So there is programmed death, the cell death 1. This is These are the molecules which they express, and cytotoxic T lymphocyte. These two are very important. These are the two signals which produce IL-10, and this produces the T regulatory cells. So I'm not trying to confuse. Just remember these four molecules, TH1, TH17, they are inflamed, they are red in color. These are going to cause fungal clearance. If T regulatory cell is good in number, it will produce a clearance and bring down the inflammation. But if T reg continues to be very high, there is no one to stop T regulatory cell production or a TH2 production, then fungal persistence will occur. So this is what the slide tells you. Next are, so this slide is just telling you, I've just taken three major examples, Candida, Crypto, and the dermoific fungi, which are the cells which are important for controlling the infection. Like if you look at the neutrophils, just one example, neutrophils can kill Candida, but it has very limited role in Cryptococcus. So in a neutropenic patient, we would rather look for a chronic mucocutaneous candidiasis than look for a disseminated Cryptococcus infection. A neutropenia patient will have disseminated mold infection, may be protected from disseminated Cryptococcus because it is not one line, the line of defense for Cryptococcus, whereas macrophages is important for all. The dendritic cell, the CD4, CD8, they are all important. So this gives you a very holistic this thing about how which are the cells which are looking after the first innate defense mechanism, and then the acquired immunity, the interleukins, a TH1 or a TH2. So what one line you must remember, a TH1 is favorable, a TH2 is unfavorable because for small time, it is favorable because it brings down the TH2, tones down. But if it continues to release IL-10, the T regulatory cell continues to produce IL-10, then it will be an anti-inflammatory response and the fungi will persist. So this is a double-edged sword. That's what I was trying to say, that a pro-inflammatory cytokine will kill. But if you have protection, if there is an impaired fungal infection, the susceptibility will lead to colonization. And if you continue to have an interleukin 10 or a TH2 response predominant, then and a TH1 response is very poor, then it will not be able to clear the infection. So it is like each TH1 and TH2 acts as a double-edged sword. Cytotoxic lymphocytes, more in viral infection, but this also causes gamma interferon and causes the fungi. In certain situation, it can also cause fungal death by cytotoxic T lymphocytes. So overall, the DC, T cell, B cell activation, macrophages, polymorphonuclear cells, complements, internuclear cellular killing. Now we come to a few slides on adaptive immunity. One is the humoral, the B cell. The B cell will get activated either by polysaccharides, which do not require a T-dependent processing. Whereas if it's a protein antigen, it requires a APC processing. It requires a CD4 cell or a APC processing when and processed, and then only a B cell gets attracted. What does the B cell do? It will cause neutralization, opsonization, ADCC, lysis, or phagocytosis. This is all very well known. But in fungi, immunoglobulins have been found to change their isotype. Switching occurs when it is a chronic infection. They produce IgE and they are not protective. They are just lying there as a diagnostic marker, but they have very little role in protection. Then comes the cell-mediated immunity. The B cell is, yes, I can only attack by immunoglobulins. And T cell says, I can attack the infected cells. And this is your lymphoid organ where the naive and the T cells are waiting for the dendritic cell to reach inside. So this is the algorithm. The naive lymphocyte is waiting, secondary lymphoid organs. Lymphocytes will require two signals to get activated, and the effector mechanism will be followed after the activation of the T cells. So overall, initially, again, this picture just tells you innate immunity, interleukin 2, It will clear by gamma interferon or a small Th1 response. Fungal elimination occurs, acquired immunity, homeostasis, and some memory cells in T cell and B cell will occur. What is the T cell doing? So T cell will have two actions. One is to kill the target, cytotoxic. Helper T cell will also stimulate and produce cytokine, and regulatory T cell will control these two. The dendritic cell, just keep a look at the picture. The antigen is now being processed by the antigen presenting cell. It is going into the lymph node. The lymph node is now giving it to the naive T cell. The naive T cell does not know what to do. It has to identify this antigen. It is not so automatic. So there are a lot of intricate signaling molecules which are occurring for the antigen to be accepted by these naive T and B cells. This is a picture of dendritic cells, very important cells. So an example where the skin dendritic cell, these are known as immature. So immature dendritic cells will enter into the lymph node. When they enter into the lymph node, they become mature. And that is the time, if you remember what I said, co-stimulatory molecules are released. Once they pop out those co-stimulatory molecules, they become the mature dendritic cells. So the first step is antigen recognition. They present the T cells. Immature becomes mature. And extracellular antigens do not require any processing. Okay, so here, now once the processing, the T cell now is maturing and trying to identify the antigen and which CD4, whether it is the CD4 or the CD8 cells, it has to get activated. And what will that decide? If you remember, what does decide CD4 and CD8? What decides the CD4 and CD8? If you remember, it is the MHC, the MHC class, which will process the antigen. So now antigen processing is when the DCs enter into the lymph nodes, their co-stimulatory molecule is one is CCR7, and they reach to the naive cells as well as T cell and the B cell. So you just look at this picture, beautifully explained that class 2 macro MHC molecule or glycoprotein is being processed within a phagosome of a dendritic cell. This is class 1, which will attract CD8. A class 2 will attract CD4. So all these cells are now processing. They are now forming a very stable invariant chains. It's a complicated signal pathway which is occurring inside, and they are preparing the antigen to be given to the T cell. Not going into the each step, because this is what the picture is telling you, that this will now stabilize the antigen into a complex which is bound to the MHC2. Now this is the step where, look at this, the MHC is now attaching to the T cell. So it is the second signal, the CD28, the CD40, these signals are now getting attached. So one pop out is the MHC antigen. This is the first signal which the T cell receives. And the second signal is this. The moment both the bullets go inside the T cell is very happy that I'm now got activated. Now it starts maturing and starts clonally proliferating. Same way by B cells, or it is by MHC. I'm just looking at this because these are the robust cycle where the intricacy of these molecules will adhere to the T cells, and then the activation occurs. So whether it is Th1 or Th2, to T cell activation receives two signals. One is the antigen, the second is the co-stimulatory molecules. And this second signal is CD8, T86. These are all, those receptors which are pointing out from the dendritic cells. So this is B7 family members, B cell. This, as postgraduates, you must know signal 1 and signal 2 is very important for T cell to fully mature, and it will start proliferating. And as I said, CTL4, these are expressed in T cells only when you want to bring down the immune response. So these get expressed that enough. Now you have done enough of immune responses. Now we need to curb down. So CTL4 will get expressed and bring down the immune response. Affected T cells. Now these T cells will now come out from the lymph nodes and they will then migrate towards the site of infection. You may ask, specific ones will go to the site, whereas the rest of the activated cell remain in the circulation. And so whether it is MHC1 or MHC2 or CD4, CD8 and the cytokine milieu, Th1, Th2 response will happen. So polarization is what it is called, depending upon the antigen, depending upon the cytokine. It can be either a Th1 or a Th2. So this is again a beautiful picture where the antigen processing is done by the dendritic cell. So these are all, card9 is one of the very important molecules in Candida. If card9, a signaling molecule in this dendritic cell is missing, it causes mucocutaneous candidiasis. You can see cases where children or monogenetic disorders where children come with fulminant candidiasis, look for card9 deficiency. After the APC has processed, then they release cytokines, and then either it is a Th1, Th2, Th17, B cell, and then T regulatory cell also tries to inhibit the immune response, either bringing down or leading to collateral damage. This is just giving you the half-life of the various cytokines which are released, inflammatory and pro-inflammatory. And last is homeostasis, regulatory. So T regulatory cells, it is not like 2 plus 2 is equal to 4, that the regulatory cell will start coming into action. So T regulatory cell has to be in large population to bring down the inflammation. If you have seen in COVID, the inflammatory response, IL-6 response was so much, so fulminant, that the T regulatory cells were there was overwhelming Th1 response. That's why T regulatory cell could not bring down the Th1. So the T regulatory cells also have to be, if the host is normal, healthy, the regulatory cells will be in enough number to bring down homeostasis and bring down the inflammation. Now I will just show you a few slides. So that covers the entire innate immunity, the acquired and immunity, which has the cellular and the humoral immunity, and which can go into any polarization, the T cell polarization. Now, genetic and immunological defects, this can be either innate. So as you said, there can be a loss of phagocytes or complement, T cell acquired immunity. Any loss can give rise to any kind of infection. The ligands, why I was telling you repeatedly about CD40, CTL4, if these ligands are absent, there will be no interaction. This CD40 ligand is very important for the APC to transfer the antigen to T helper cell. If this is not there, the T-dependent antigens will never reach the T cell. The T cell will never get activated. That's why these ligand, now there are panels where the immunological panels are looking at these markers and trying to find out where is the deficiency. So immune defect, if there is a neutropenia, what kind of infection can you think? Neutropenia can be after hereditary or any genetic Cosman syndrome. They will acquire Candida, Fusarium, Mucor, dimorphic fungi. I said Cryptococcus is one thing which you can keep out of your mind. Second is a functional defect. Suppose a chronic granulomatous disease. This is a functional defect in the neutrophil. The number of neutrophils are okay, but the phagocytic activity is poor. That means poor reactive oxygen. So oxidative killing is poor. Leukocyte adhesion deficiency. I told you about the endothelial adhesion molecules. If those molecules are deficient, the leukocytes will never reach the site of infection and will never cause phagocytosis. CHS is another case where they form bizarre phagolysosome. They cannot phagocytize and the fungi can remain. Abnormal phagocytosis, myeloperoxidase deficiency, and functional defects, even in diabetes, HIV, steroids, these also can cause neutrophil defects. Molds, Mostly neutrophils are the first line, but macrophages are the biggest, the bold killer line of defense. Conidia will escape the first line. If they germinate, hyphae has to be captured by the neutrophil and the macrophages. So the dendritic cells have to activate the B and T cells, and the same procedure. The moles usually get activate a Th1 response or a Th17. Candida is mostly Th17. Moles usually have a Th1 response. If you remember the post-COVID COVID mucormycosis, there were the host protein that was the GRP78. These were the PRRs which were found in a paper where they found the host were expressing in the alveoli. They were expressing the GRP78 in large numbers. And the mucor, which has cottage, is their PAM molecule. They were also in large numbers. So both ways, the TLR, the PAM as well as the PRR, there were so many in number. That's why they caused a fulminant mucormycosis in those post-COVID patients. A Th2, if suppose there is an anti-inflammatory response, there is no one to defend, it will cause invasive aspergillosis. High IL-10 is a signature, is a sign for progressive invasive aspergillosis and even alveolar allergic bronchopulmonary aspergillosis if there is a high IgE and interleukin 10. Candida can be either acute disseminated candidiasis, mostly seen either in the GIT translocation, like after malignancy in chemotherapy, or through IV catheters. It can manifest as chronic manifestation or a disseminated, as I said, in CARD9 deficiency, chronic mucocutaneous also in CARD9, and disseminated can be after suppressive therapy. So this is the red ones if you see. The reds, CARD9, NLR3, DOC, STAT. These are the signaling molecule. Any patient having these missing, the signaling molecule will not work. Antigen cells will not be presented to the T cell, and the patient will have fulminant infection. So see, CARD9 is a very important factor for dermatophytosis as well as candidiasis. They have poor Th17 and a Th1 response. There is a allelic mutation which occurs and causes colonization of invasive candidiasis. Autoimmune polyendocrinopathy candidiasis, this is again a very rare AIR gene mutation causing a triad of candida and other organ disorder. So this is again a table showing you. So don't think, see, look at this. Chronic mucocutaneous, invasive dermatophytosis, all is because of genetic disorder. Again, genetic disorder. Lot of candida infection. Cryptococcus requires macrophages. The capsule is protective. Variants, histoplasma, again, principle is macrophages and acquired. And so I'll just briefly, just two more slides. This was a test that we did, a small study where candida was taken from normal mucosa, some candida which harbors in the oral mucosa, and one which was taken from patients of diabetic. Their polymorphonuclear cells, PBMCs, were stimulated with candida. One is arising from healthy control, the other is arising from the diabetic patient. And what happened? The diabetic ones had a high IL-10. They could not mount a gamma interferon response. Whereas a normal patient, the healthy controls had a higher gamma interferon. Their neutrophilic activity was normal. So you see, when a candida is in a chronic state, it will produce some kind of, suppose a glucan or a mannan, which is getting identified by Th2 response. So there is IL-10. So diabetes usually have this deficiency of mounting a good Th1 response, unlike the healthy controls. And in dermatophytes, again similar, what you have seen now, chronic dermatophytosis, they have found they are either associated with deficiency, or there is again a switch towards a Th2 response or a massive Th17 response. And these are beautiful pictures which, when you go into higher studies, then you will try to understand what is this complication. The Th1 is trying, The TH17 is trying to block TH1. So there is a fight between TH1 and TH17. Both TH1 is high, TH17 is also high. Now who wins? Ultimately, the TH17 wins. But to stop the TH17, the T regulatory cell also has to work. The T regulatory cell is at a loss. So in this picture, you see the T regulatory was unable to stop TH17, and this is what has resulted in chronic infection in dermatophytes. So these are very simple pictures which you will understand on your own. This line means inhibition. So T regulatory cell is trying to bring down TH1, whereas it is unable to clear the TH17. This is very robust. So there is a chronic inflammation. Fungal clearance, TH2 is trying to clear the fungal clearance, but TH17 is very high. So you remember the first picture, the orchestra. So what happens is everyone is playing their own instrument. So there has to be a control. Any of them becoming, trying to be out of tune of that interaction, then it causes a lot of damage to the host. This was what I was talking about, mucor. Finally, mucormycosis dissemination. And here I'm telling you, don't get scared by another cell. This is a myeloid-derived suppressor cell. This is again a cell which a progenitor cell which was studied in mucor, that they were very high and they are the ones which caused inflammation. So these are, I'm saying that immunology doesn't end here. There are newer cells. I will show you another picture, innate lymphoid cell. These are again a very newer lymphoid cells, progenitor cells in circulation, which have a very good activity in the innate immunity. So immunology never ends. This is what I've told you is very basic concept, and intricacies you keep on learning the cells and their mechanism. It becomes very clear when you look into the pictures. So these are the references, and I'm ready to take any questions. 1 minute. Oh, 1 minute.
Thank you, Dr. Shukla. That was made so easy and seemed so simple, but we all know how difficult it is to understand. TH1, TH17, TH2, it's all very confusing, and you made it sound so simple.
I think I stopped in time. I thought I will go beyond because over each slide you can take about 10 minutes to explain. But I think these, if you only, at our time, I think I remember we used to read riots and only look at the pictures. You don't have to look into the theory. So the pictures are so very well explained, and you will be able to understand the immunology. So if there are any questions, otherwise.
Right. I can't see anything in the chat box. There are no questions in the chat box. If anybody wants to ask something, please unmute yourself and ask. I think Bharti ma'am is traveling. I think she could not join us today. So we'll wind up. Thank you so much, Dr. Shukla, for such an informative and a topic which is so difficult, made it so simple.
Okay.
So I think we had all the accolades for you. Everybody praising the lecture only in the chat box.
Thank you so much. I
so
tried
much.
to make it simple. Okay. Thanks, Dr. Sonal, for this opportunity, because every time you read, you enjoy and then you understand it and then explain. So I hope the students have enjoyed it.
Right.
Okay. Thank you so much.
Right. Thank you so much. So I'll end this meeting.